Good evening, everyone. It's a pleasure to be here, and many thanks to the Royal Society for the invitation to present this work to you. This is, in fact, something of a summary of a very extensive volume of work that we've done on hyper-arid and other desert soils, hot desert soils, over the last nearly 15 years. And although it's titled The Survival of Microorganisms in Hyper-Arid Desert Soils, in fact, it's rather more than that. In fact, it's the life or aspects of the life of microorganisms in these extreme desert soils. So I've just listed, there's a whole bunch of co-authors there, people who've contributed in so many ways. Not all of them in my group. A couple of the photographs, particularly the bottom left, that's Belinda Ferrari from the University of New South Wales, and her name will come up again as we proceed. So there we are. So this is the Namib. For those of you who don't know Namibia, the Namib Desert is a western coastal desert. It's approximately 180 kilometers from the coast, and the left-hand end of that arrow is in fact in Walvis Bay town, and Windhoek is about 220 kilometers to the right-hand side of the right-hand end of the arrow. So that's to give you a sense of scale. So the Namib Desert is a particularly interesting desert. It's nearly unique in the world and it's unique in the way it acquires water. And as you'll see as I go along, water is a key in desert ecosystems, obviously. So the only other desert where you find this would be the Atacama. It has a fog zone. The first 60 kilometers is inundated from time to time by fog. And fog, of course, comes in from the ocean. Those of you who spent time in Walvis Bay will know that the mornings are typically miserable because it's cold and foggy. You drive an hour inland and it's extremely hot and dry. But so the fog zone is the first 60 Ks. Then there's an intermediate zone, which we term the hyper-arid zone, where there's typically no fog at all and very little water of any sort. And then on the eastern side, we have what we call the rainfall zone. Well, this is all relative. It is a higher rainfall zone. So we have this gradient of water availability. It is a gradient, it's a transition. It's not a zonation, that's our own artificial structure, but it works it's quite a useful thing to apply. There's a transition of water availability from the coast to the eastern edge of the desert. And that makes it a particularly nice study system to try and understand the way in which biology in general, and microbiology in this particular case, is influenced by water availability and the other abiotic parameters that typically influence soil systems. Now, for those of you that know the Namib, you'll know that it's a desert of two halves. The northern half, you're seeing most of it in this particular slide, is a gravel desert. This is a typical gravel desert vision. Desert pavement, so covered in small pebbles, very mixed geology. Although you will note, and I will mention them briefly in passing, there are a lot of small white rocks in this particular shot. Those are small quartz pebbles, and quartz turns out to be particularly important. But this is essentially a hot, depauperate desert with typically very little obvious or overt biology. The southern portion, which goes from the Kasib River all the way down 500 kilometers or more down to the South African border, is the Great Sand Sea. This is sand dunes, magnificent sand dunes with interdune zones. And these in many ways are different. We study typically the northern part, the gravel deserts, that we have done some work here, which I won't be referring to, but both have their own unique biological systems. And some of those unique and interesting systems are just shown, for example, in this slide here. So just a quick summary without any detail of some of the more interesting parts of the biology of the Namib Desert. From the top left, we have, of course, the free-living, the microscopic, the free-living soil microbiome, about which I'll say rather more as we go through. Then there are some unique structures that harbor discrete elements of that microbiome. And this, in the second slide from the left on the top, these are rhizosheaths or rhizospheres, incrustations, mineral incrustations held together, as it turns out, by microorganisms around root hairs, a very unique and interesting system. Then we have biological soil crusts with some lichens. In the moister areas, the western areas, you can find biological soil crusts with lichens and other systems attached. And then the one I will talk about to some extent towards the end of the talk, the so-called crypto hyperliths. A hyperlith simple hypolith means under a rock. Quartz rocks, if upturned, have this green incrustation on the underside, and that's a unique, cryptic, and very interesting community, the so-called biological hotspots of the Namib Desert. Then you have lichens. The famous lichen fields have been studied for years by many people on the coastal zones. And then in the bottom row, just a selection of pretty slides of other things, which have been the subject of study by botanists for many years. The euphorbias, the famous enigmatic Welwitchias, some very interesting endemic xerophytes that somehow scratch out a living in the most difficult parts of the environment. Hoodia, of course, in the eastern zone, the famous supposedly appetite suppressant plant, which maybe isn't. And then, of course, I'd put in the last slide there what I've called transient succulent annuals. When it rains, and that's pretty occasional, the desert changes, as you will see. So one of the many things we've looked at, and one of the things that microbiologists, and for that matter, any biologist is interested in, is what are the drivers of community structure and community function? What are the main effects that drive the way in which the community assembles and the way in which it functions? And I list in no particular order on the left-hand side the dominant drivers in this system. Light, of course, for photoautotrophy. Temperature, this is a hot desert, as you'll see in a second. Water, it's a desert, and water, therefore, is vitally important. And then the other, possibly less critical, but still significant drivers, the oligotrophic state or nutrient availability of the community. UV radiation is high in desert systems, clear skies. But remember, UV radiation, although we note it as an extreme condition, you can hide from it very easily by living under a sand grain. Physical substrate availability is important for some species and maybe not for others. And then oxidative stress, generally under-considered and closely related to water status. But in a dry environment where there's very little water, then oxidative conditions dominate, and oxidative stress is in fact a really important element of adaptation in these organisms. I said it was a hot desert. Here is a roughly year-long temperature, surface soil temperature plot taken from eye buttons, which we use routinely. And peak temperature in this particular year, I forget which one, somewhere in the last decade, surface temperature is 66 degrees centigrade. They go higher. We have measured up to 72 or 73 at the surface. Remember that egg protein of albumin starts to denature and coagulate at about 62 or 63. So this is hot enough to boil an egg. And the mean temperature, mean summer maximum, 58 degrees. So this is a genuinely thermal environment. And you might think that temperature is a very strong driver of the biology in the system, and particularly the microbiology. But in fact, I argue that it isn't. And I'm going to put temperature aside largely and look at other features. And the main feature, because it's a desert, obviously, water is everything. And this is a photograph taken of the gravel desert somewhere in the center of the hyper-arid zone in April 2010, after somewhere more than 3 years of drought. I think 5 or 6, since they'd had significant rainfall. The following year, and we have been going there on a field excursion for a week, every April since 2010, it had rained. And the desert had become a prairie. This is roughly in the same zone, not quite looking the same direction. And that is massive growth of Stipagrostis. It's one of the grass species. So for a while, it was a transition from a genuine depauperate desert to a highly productive desert. This is absolutely typical, of course, after rain, but rain is a very, very infrequent event. This was, in fact, that they had 70 millimeters of rain over about a 2-month period, which was a 70-year rainfall maximum. And they've had very little rain in that zone ever since that time, just 1 or 2 falls. But so water is everything. So we have focused on water as a driver of biological systems that we look at, which are the microbiological systems. There is water there. So we have dug shallow pits and implanted eye buttons. Eye buttons are little monitors, little, they look like buttons. Very useful little monitors that will record temperature and humidity for long periods. They will hold 6,000 data points. So you can leave them and sample every 3 hours for an entire year. And we've done that. And here, these are eye button humidity, that's atmospheric humidity or soil atmosphere, humidity data from a shallow pit down to 20 centimeters. And if you look at the top, on the surface, the atmospheric humidity is about 20% RH, or put another way, very, very dry. But at 20 centimeter depth, we've got a soil atmosphere at 96% relative humidity, near saturation. Now, you've got to be careful with relative humidity. It doesn't necessarily reflect the moisture in the soil. And if we had looked at gravimetric soil content, water content, we would have still rather low figures. At the surface, water content would be about 0.15% by weight. A moist garden soil will be 5 to 7%, just to give you a sense of scale. But there is water there. And so one of the questions that we've been asking is how accessible, where does this water come from? Because in central Namib, it may not rain from one year to another. And how accessible is it to the microorganisms that live within that soil profile? And this is going to be a theme for the next 10 minutes or so. So here we have a range of the different options that we consider as inputs of water to the system. From the top left, rainfall, obviously. Rainfall, it's a very infrequent event in most parts of the desert. So although it has a massive impact when it happens very, very rarely. But it impacts on all the surface and subsurface features and biological systems. And along at the interface between the soil and the sky, if you like, I've drawn in figures. The biocrusts, which are surface growths of lichenized material, algae, cyanobacteria. The next one is my favorite, hyperlith. That's a quartz rock with this green cyanobacterial scum on the underside, embedded in the desert pavement. And then I've also listed the crustose lichens, which occur on little rocks, which are rarely as neatly shaped as that, but only in the coastal area because they are fog-driven. So along the top, we've got rainfall as a very, very infrequent and therefore not a dominant effect in terms of water input. On the right-hand side, we've got fog deposition. Fog is limited, as I showed right at the start, to the first 60 kilometers and generally rather less than that. So in the central Namib, fog is not a water input, and we have to be looking for other sources. Dew condensation is a possibility, but to get dew, you need to get the temperature of the surface below the dew point. And given that may happen from time to time in the winter months, but it's certainly not common, the dew point will rarely be reached in the summer months in the central Namib. So dew is probably not significant. And then you'll see in the middle there, so-called nocturnal distillation. Once again, this is essentially a condensation process, but this time the water is coming from soil water, which is essentially, because of the thermodynamic driver, from a high humidity below the surface to a low humidity above the surface. There's a driver to move water vapor upward and into the air, which will then condense if the surface temperature is low enough. But as I said, it rarely is. And then on the right-hand side, although it's partly obscured by pictures and names, so we have nocturnal recharge. Now, this is a new concept, or at least new to me, which I know is being studied actively by research groups in Israel and elsewhere, where essentially soil atmospheric humidity, because the atmosphere, because of the cooler temperatures at night in the desert, is moister than during the day, and the soil remains very dry because of the heating and evapotranspiration during the day, essentially recharges by absorbing moisture from the atmosphere at night and then giving it back during the day. That has not been quantified yet, but it's very interesting. There are two other possible sources of water, one which nobody else really thinks much about and which I need to understand better, and that would be water vapor from deep groundwater. Now, groundwater, the depth varies across the desert. In places it might be 20 meters below the surface. But nevertheless, the fact that the humidity, the subsurface humidity of the soil is high, suggests there's a possibility of the slow transport of groundwater from deep subsurface to the surface, driven by the same thermodynamic drivers, the difference in humidities, and the question of whether that water vapor, or for that matter, the water vapor that comes from nocturnal recharge, is accessible to the microbiome is still one that hasn't been answered, and is a very good question. There is one more, and I'll come onto it at the end, and it's on the left-hand side of the screen there, which is metabolic hydrogenesis, the capacity for organisms under some conditions and with some substrates to make their own water. And that's a very exciting open question at this point. So we know that there's water in the soil. I showed you before, and here's another graph that shows that. And this is a 7-day I-button soil humidity plot. The blue lines, that's the surface humidity, and you can see there are massive changes throughout the 24-hour cycle. Now, I remind you that relative humidity is actually a temperature-dependent variable. And so if these were normalized for temperature, and they're not, they wouldn't be so dramatic. But nevertheless, the suggestion is that at or near the surface, organisms, because assuming they're in equilibrium with the atmosphere around them, will go through hydration, dehydration cycles every day. And that, of course, is particularly important in terms of their physiology and their metabolic capacity, which I'll come on to. The orange line shows the same profile at a depth of 10 centimeters below the soil surface. And although there's some interesting lag effects, which I'm not going to discuss, you can see there is substantial buffering. There will be both thermal buffering and humidity buffering. And I didn't show it, but the 30 centimeter line or the 20 centimeter line would show relatively low variations on a daily cycle. So the point here is that organisms at or near the surface, and the ones we study are generally in the top 2 centimeters, are theoretically subject to very large changes on a daily basis in cellular water content. And that has some interesting physiological implications, as you'll see in the next slide. So this is a very complex slide that I'll just pull out the key elements. This is essentially what happens, and that's the line that dips in the middle and climbs again, to an organism when it dries out and then resaturates. These data come from all sorts of sources, from work mostly not done on microorganisms, some on tardigrades, some on resurrection plants, including from Jill Farrant's lab at UCT and others. There's a lot of work behind this. And so the absolute tipping points, if you like, which I'll talk about in a second, are subject to some questions, and they're certainly not finally resolved. But nevertheless, as you go from a saturated state to a dry state, which as suggested in the previous slide, might happen almost daily for some organisms, the first thing that happens as water leaves the cell, as the cell becomes desiccated, is that you lose the higher order functions. Possibly, in the first drying phase, as the water activity drops from 1, which is saturated, down to maybe 0.9, you lose cell division, you lose motility, so the high order functions. Rapidly thereafter, as you go down to maybe a water activity of 0.75, you'll lose some of the other key metabolic processes, carbon fixation, nitrogen fixation, for example. As the cell dries, things happen. There are a series of response processes in response to cellular desiccation, which upregulate gene transcription, switch on pathways, and particularly pathways designed to produce products, compatible solutes, so-called, which will protect the cell in its dry state. So, and this comes from many organisms, including microorganisms, switching on synthesis of sugars, sugar alcohols like mannitol, for example, trehalose, disaccharides, things that pack the cell and prevent damage to cellular systems. So those upregulate for a while until you get to a water activity of about 0.5, and then we think that most of the metabolic systems shut down completely. As a cell dries further, there's relatively little happening. And this is a phase, maybe we've defined it as below water activity of 0.5, as a state of anhydrobiosis, which is a term that's been used for many, many years. In other words, essentially living in the absence of water, although it's not in the absence of water. There's always some water retained in the cell. And one of the questions that's really not been answered well is, what is the state of metabolic activity in the cell under these very dry conditions, so-called anhydrobiotic conditions? I'll have some answers to that later in the slideshow. Then, as water becomes available from whatever source and the cell resaturates, the reverse process happens. Transcription switches back on again, metabolic pathways are activated, certain things are downregulated. You no longer need to synthesize compatible solutes. Stress response genes typically downregulated. And at some point, you recover your higher order functions. Exactly that point we don't know about. So this is what happens in a cell. And we've defined, as have others, the amount of time that the cell sits in a condition where it can have some of these functions. We call that the metabolic window. So what we've attempted to do is to take these tipping points or cutoff points for various metabolic processes and overlay those with the water activity conditions, which we get from relative humidity, in the soil of the Namib Desert, to try and work out how much of the time the cell is actually performing these critical functions. So, in fact, comes from this particular very messy slide, which is a full annual cycle. In a year when it looks like there could have been 4 rain events, which is quite unusual. But if you look at the red dotted lines, I've defined the point at which, at 75% R rate, at which nominal higher order functions disappear. That's cell division, motility, and nitrogen fixation, and maybe carbon fixation, but certainly nitrogen fixation. And then at 50%, I've defined the transition to the anhydrobiotic state. And so we've essentially just mapped that graph against those parameters and come up with some numbers. So microbial growth, which is the first to go, cell division, and this is in the hyperarid zone, in a shallow surface soil. We estimate that a cell is capable of cell division only about 5% of any average year. Obviously varies year to year, but these are just examples of what might happen. Photosynthesis, fractionally more because it may be slightly less sensitive to drying, but still only 7% of the year. Amino acid metabolism, which is certain parts of which are defined as part of basal metabolism or basal energy metabolism, possibly functioning for much longer, but this is part of the anhydrobiotic state, if you like. And the differentiation between amino acid metabolism, basal metabolism, really quite hard to define with the knowledge we have at the moment. In fact, some of the information comes from my master's thesis circa 1971, so we won't go there. But nevertheless, it provides fairly clear indication that higher order functions are highly constricted. The metabolic window for higher order functions and the ones that actually provide the ecosystem services for the microorganisms in these soils in hyperarid deserts are very, very limited. So now, with respect to that, I want to talk about some of the work we've done to actually try and investigate what metabolic systems are functioning in the microbiome and what elements of the microbiome under different water conditions. And this experiment was performed by JB, longtime postdoc who's now in Chile, Carlos, postdoc, and Pedro, senior postdoc who's still with me. So this was an experiment where we used metatranscriptomics. That is, we isolated total RNA, fractionated out the messenger RNA, and used mRNA as an indicator of what genes were being expressed, and therefore what pathways were functional. And it's a very powerful method, very data-heavy method for looking at metabolic community metabolism, because you can drill down into the question of what pathways are functioning and which organisms have those pathways functioning. So it's extremely powerful. this particular experiment was designed to look at how functionality in dry soils, this was in the hyper-arid zone, very low water content, how they varied over diurnal cycles. So we were attempting to address that issue of nocturnal recharge, although we didn't know it at the time, but I'm not sure it showed very much. So the question was what genes were functional, and therefore what pathways are functional, and what taxa were involved. And I've got a single slide to summarize a large bulk of work from that. And it's quite revealing in one way. Firstly, on the left, we've got a summary of the genes involved in nitrogen cycling. And what it says essentially is that nitrogen acquisition by cells is still active using inorganic nitrogen in the form of nitrate. Nitrates, in fact, are quite common in deserts. In some deserts, in places like Chile, that the concentrations are so high you can mine them for fertilizer. The nitrates are high in the NAMib, but not at that level. So it would appear that nitrate reductase and nitrite reductase, the two enzymes that take nitrate to nitrite into ammonia, where it can be assimilated into organic materials, and particularly into glutamine with glutamine synthase, those are active. They were the dominant processes. And so nitrogen sources essentially driven by inorganic sources. Dinitrogen fixation, or nitrogen fixation, gaseous nitrogen fixation, virtually non-existent under those conditions. And that fits with what we would expect, because nitrogen fixation's got a very high cutoff in the desiccation process, above 75% RH, or around about that point it probably cuts off. And this is a dry soil, of course, at much lower water activity. So that fits. In terms of carbon cycling, on the right-hand side, carbon cycling or carbon uptake is essentially heterotrophic. So the inputs there, glycerol 3-phosphate and glycerol, these are essentially products of phospholipids, and therefore almost certainly plant phospholipids. So these phospholipids derived from plant detritus from one of the previous growth periods, in this case, some years earlier. But since grasses grow, you saw the photograph earlier, they degrade, they fragment, they get incorporated in the soil, and they become available, all the elements become available as heterotrophic substrates for microorganisms. And so it appears that under these conditions, the principal carbon uptake is using the products of plant phospholipids and pushing them into the cell into glycolysis. And interestingly, although we don't really understand why, driving inorganic phosphate uptake. We've seen phosphate as a driver of community composition in other studies and never really understood why. But it would appear that in order to be able to assimilate glycerol, you have to phosphorylate it, therefore you need phosphate, therefore you must take it up from an inorganic external source. And it's interesting, the phosphate transporter, cell membrane protein, is very highly upregulated. The other interesting fact from this, and I'm not showing you the data, is that only a small subset of the microbiome were actually active. So when we looked at the phylotypic affiliations of these genes, and they're not shown here, just a small fraction of the microbiome was functional, and the rest, the large proportion of the cells and taxa in the soil, and this is a very complex community, were dormant. And that would fit, I guess, with our current understanding. What is, looking at it the other way around, though, it's very interesting that some of them, as shown from these data, were not dormant. They were maintaining some functionality. So then we asked another question, which was, okay, we now know what's happening to some extent in very dry systems when there's very little water available. What happens when water arrives? We tried an experiment waiting for a year for it to rain, and it didn't, so we gave up on that. And so we decided to artificially water a piece of desert, that's shown in the picture here. This is after the experiment was complete. The so-called watering can experiment. We used ultra-pure water, so no DNA background signal, so we weren't confounding the microbiome DNA signal with what we're adding in the water. We used artificial rainwater, which has a bit of salt in the background. And we watered a piece of desert, and once again used metatranscriptomics, that's RNA, messenger RNA-based sequence analysis to try and understand who was functional, who was responsive, and how were they responding. This summarizes it. So the top left-hand side is just to show that when you add water, that's the blue bars. When you add water to soil, it gets wet. That's the summary of that one. The right-hand side figure with the dots is, however, fundamentally interesting. So within the dotted circle, we have each dot represents a composite metatranscriptome. That's all the RNA expressed, or mRNA expressed from all the organisms. And if the dots cluster together, it means essentially those expression patterns are similar. So within the dotted circle or oval, they're all broadly statistically similar. The blue dots at 10 minutes, and then subsequently the blue, green, and light blue dots represent metatranscriptomes at 10 minutes, 3 hours, up to 1 day, 3 days, or in fact, in 7 days is back inside the dotted zone. So even after 10 minutes, the composite metatranscriptome was fundamentally different. Huge changes had happened just in 10 minutes. And those changes continued throughout the period up to 24 hours. But note, by the time we get to 1 day, we're already starting to look like the basal or control system. And at 7 days, we're back where we started. Everything is shut down again. So first take-home message, 10 minutes and response, massive response in the expression levels, the gene expression levels of the organisms in the soil. And when we looked at the identity of those genes and what they were encoding and the pathways and systems that they were representing, it was exactly what one would hope for in a way. So I summarized below there the so-called wet genes, those that were significantly upregulated by water addition as quickly as 10 minutes. Motility, a whole series of motility-related genes. And this makes perfect ecological sense because when you add water to a soil system, you provide a liquid continuum. Prior to that, cells are essentially stuck to soil particle surfaces as biofilms. They can't go very far. They can't move to new substrate areas. Whereas you add water, you provide a liquid continuum. And so it makes sense that a cell would switch on its flagellum, its motility capacity, so that it could move through that continuum to a high substrate zone. Having got there, it's got to be able to use the carbon that it finds. And so import, particularly transporter genes, were really significantly upregulated. I'll come on to the secretion elements in a second. Also, not surprisingly, when water is available and substrate becomes available, you switch on the cell division machinery. The objective of one cell, of course, in the microorganism world, the only objective of a cell is to become 2 cells, and this is what they're aiming to do. But the other thing that's interesting is that there are 2 genes, the type 4 pili and type 6 secretion systems, or genes for those, which are upregulated, and both of these are implicated in all sorts of intra-species and intercellular competition systems. So once cells are mobile within this continuum, they will be encountering other cells, other species, other taxa. And so competition, methods, systems, biochemical processes in order to combat other organisms would appear to be a significant factor. Enhanced competition, it makes perfect sense, in fact. On the contrary, the so-called dry genes, those that were downregulated when we added water to the system, also fitted with what you would expect in ecological terms. When you add water, the system becomes less aerobic, and cells are subject to less desiccation stress and less oxygen stress. And so we found that quite a lot of the stress response genes, the chaperones, HSPs, those are heat shock proteins, DNA-K, very well known stress response gene, these were downregulated. So no need to protect the cell to the same extent. And the other thing that was downregulated was essentially autotrophic carbon fixation. That is rubiscos, which are genes in the carbon basal cycle, particularly in photosynthesizers. Autotrophy is an expensive process in energetic terms. And if you've got an abundance of organic carbon around you, such as you might have once you wet the system and free things up, as it were, you don't need to spend a lot of energy on autotrophic carbon fixation because you can get it by simple adsorption. So it was a very nice experiment. It produced some data which was entirely consistent with our predictions. And we are now, in fact, in the middle of an experiment which is essentially the same. We're going to duplicate it, but this time our first sample point after water addition is 45 seconds from water addition. And those metatranscriptomes, we've isolated the RNA, and those are being sequenced at the moment. So within the next 6 months, we will know even more how much more quickly things respond. There's also changes in cell growth, and this is an extremely complicated and messy slide. There are really only two messages. On the left-hand side, you've got dry and watered. The dry is control, nothing much happens. These points, these reflect ribosomal protein content, and it's some sort of proxy for cell growth. So you can see on the left-hand side, there's a bunch of taxa, each one with its own colored lines and dots, which appear within 10 minutes to have already vastly increased ribosomal protein levels, and then they're tailing off, as it were. So they've gone through a burst of growth inside 10 minutes. Those would be the early responders. On the right-hand side, you've got the later responders. So at 10 minutes, a bunch of them, look at the delta proteobacteria, they're still rising up into 7 hours to 1 day in the case of the oligohymenophoria. And the interesting thing about these taxa is that these are, certainly some of them, predatory organisms or predatory bacteria. The delta proteobacteria contain a range of predatory bacteria. So it makes sense if your objective, if your substrate is another bacterium, it's probably wise to wait for the cell numbers of your substrate to increase before you start increasing your own cell numbers to take advantage of it. So it was an interesting experiment, and I think we need to look at this in much more detail. Through all of that and from all of the data, we came up with a sort of model for what happens, and I just show a picture here because postdoc did a brilliant job putting this together. When you add water, as you can see, the blue area surrounding the cells, you have a liquid continuum, and it triggers the stimulation, motility, growth of a range of things, including viruses, which I haven't talked about. And then you start quite quickly, 3 to 7 hours, the process of redrying. Remember, these are surface soils. They're subject to the extreme heat of the midday sun in the Namib Desert. So drying is fast. And 3 to 7 hours, we have the myxobacteria, the predatory bacteria out from the delta proteobacterial group, some eukaryote predators, so for example, amoeba, all starting to increase once the cell numbers of their substrate have increased. And then by 1 day, much of the water is gone. The liquid continuum has probably disappeared. You now have a biofilm of water around the soil particles, and systems are switching off. Other systems switching on, so heterotrophic substrate acquisition is probably diminishing, and there's a switch on of autotrophic processes, so carbon fixation from CO2. And then finally, after 1 day, you're back to where you started. You're in a dry system. Some organisms have switched off, some will have sporulated, some will remain some degree of activity. So it makes a nice continuum. I'm now going to talk briefly about the continued issue of where the organisms get their energy from. So the standing paradigm in systems, in all systems essentially, or most systems, is that photoautotrophy drives the world. Plants in particular, and even in the Namib Desert from time to time, there'll be plant photosynthesis, but much of the time there isn't. In the absence of plants, you have photosynthetic bacteria, the cyanobacteria. Some of you might have known them once as blue-green algae. They are bacteria, and they are photosynthetic, of course, and although not so common, the chlorophytes, that is the eukaryotic but photosynthesis, single cellular photosynthesizers. So certainly cyanobacteria are common in parts of the Namib Desert soils and in the niche communities. The other option for autotrophic acquisition, and I'm not talking about heterotrophic substrate acquisition here, that goes on all the time, would be chemolithoautotrophy, the use of reduced sulfur, reduced iron, for example, largely doesn't occur in the system like this. But the new paradigm is this, trace gas chemotrophy. And this is a new concept, at least at the trophic level, although systems have been known for many years, which may be a really important driver of energy acquisition and of carbon acquisition in these systems. So this is what it's all about. Hydrogen assimilation, atmospheric gaseous hydrogen assimilation. Now, hydrogen in an average atmosphere is at about 10 to 15 parts per billion. That's very low concentration, but it's enough to be utilized by these high affinity type 1 hydrogenases. These are a new clade of assimilatory hydrogenases, enzymes in the cell walls of bacteria, of some bacteria, that actually bind hydrogen and oxidize it. And oxidizing hydrogen, of course, is a very energy efficient or energy acquisitory process. You get a lot of energy from it. Carbon monoxide dehydrogenases, they will equally bind carbon monoxide, which is generally a little bit higher, maybe 40 or 50 ppb in the atmosphere, and you can oxidize with some energy gain carbon monoxide to carbon dioxide. And the working theory is that the energy from these two processes that often go together, as it turns out, drives some very high affinity CO2 fixation processes. There's a new Rubisco, a type 1e, which seems to have a very high affinity for carbon dioxide. And that may be the key process here. So this work, as a trophic structure, a novel trophic system, came out of some work by a couple of Australian researchers both of whom we worked with, Chris Greening at Monash and Belinda Ferrari at New South Wales. And the work originally came from very remote, very extreme Antarctic soils where there were no phototrophs. There were no cyanobacteria. And so there had to be another source of energy, and they found these systems were functional there. But now it seems, and we've tested in our own hot desert systems, that possibly this new trophic paradigm is actually ubiquitous worldwide. It's just in rich grassland soils, for example, hidden behind a much more massive photoautotrophy, so you don't see it. There are a bunch of interesting bugs that appear to do this. There's actinobacteria, well known, but a couple of new phyla, of which there are, I think, only one isolate so far, but it does look as if it's more widespread than originally thought. So these, the papers that came out of that, the original one, atmospheric trace gas supported primary production, that was the one that hit the headlines. And then subsequently, we worked with Chris and with Belinda to look at Antarctic soil metagenomes and hot desert samples in Australia and in South Africa, and discovered that this process occurs in all those and probably in others. And in fact, it's dramatic. These are hydrogen uptake in dry soils, not in wetted soils. These are done on little microcosms, so 20 grams of soil in a bottle with a hydrogen headspace, and then you measure the hydrogen uptake by taking gas samples out and analyzing it by gas chromatography. And you can see that the rates vary. The different colored lines refer to the different sample sites, and there's something interesting in there, but no time to go into it. But if you look at the left-hand side, there are a couple of sites there where even in the dry soils with very little water available, the sort of soils we studied earlier, the hydrogen uptake rates are dramatically fast. And although it's not shown, if you sterilize the soils by autoclaving them, you get an absolutely flat line. So there's no abiotic hydrogen uptake. This is a biological process. The hydrogen provided in the microcosms is a long way above the atmospheric level, but the point is that these organisms will pull the hydrogen out of the atmosphere to below normal atmospheric levels. That I said 10 to 20 ppb. They'll go below that. So this is very exciting. In wetted soils, they get much, much faster. The systems are much more switched on. The rates are very much faster, as you can see. And that's maybe not surprising. What is probably surprising is that some of the samples were so fast in the dry soils. What are the implications of this? Apart from the energy capture, there's another one. This is an equation that all of you will have known since you were probably 5 years old. Hydrogen, oxygen produces water. But the physiological implication of this process in the context of hydrogen assimilation and oxidation in a cell is particularly interesting. Is it relevant? We don't actually know yet, but the concept is that hydrogen aerotrophy or hydrogen assimilation and oxidation might contribute to the water budget in the soil and in the cells. We've done some back of the envelope calculations, and there are small but significant additions per gram soil per day from this process using the rate data that we've got there. Whether this is of physiological and community significance is still an open question, but it's a very exciting one, because if the cells can produce their own water and stimulate their own hydrogen uptake processes because of it, you have a system which is no longer dependent on external water addition. And that would be very exciting in an ecological sense. To finish off, I'm going to talk briefly about time, because although we've done lots of qualitative and some quantitative analyses, we know nothing about the nature of the timescales and the rates of processes in many of the communities. And this particular community, you'll see on the bottom left-hand side there, this is a quartz hyperlith. I've pulled a small quartz rock, maybe 4 centimeters square, out of the soil, upturned it, and you can see adhering to the underside on its ventral surface, a nice green layer of cyanobacterial-dominated hyperlithic community. It's a biofilm. It's all bound together with extracellular polysaccharides, so it binds a lot of mineral substrate as well, but it's a classic crypto-hyperlithic. That means difficult to see. You don't see any evidence of this when you look at a quartz rock embedded in the desert surface until you pull it out and turn it over. So we ask the question of how quickly do these communities actually develop, because nobody knows. Now, I've always had a bit of a concern that when I pulled one of these out of the soil and popped it in a plastic bag to take it home to study, I was destroying a community that might have been there for 100 years or 500 years. These deserts are very stable, and the desert pavements are very stable. So we set up an array, and you can see it in the bottom right-hand corner there, of natural quartz, which I'd taken out and cleaned and sterilized, and artificial tiles, some quartz, some marble, and so-called black rock controls, rocks that have no light transmission. The characteristic of the quartz, of course, is that it allows light to pass through. It transmits light to the lower surface, which is why the photosynthesizers live there. So we set up 3 of these arrays, in fact, in 2015. And the only exciting thing about 2015 is that this was set up as a 9-year experiment in an academic environment where funding only lasts for 3 years, and where I was due to retire before the end of the experiment. So that's academic optimism for you. However, it worked. And we actually uplifted for study the samples in 2022. So that's a duration of 7 years from the initiation. And you can see on the lower right-hand side, one of the tile arrays, after certain samples have been removed. We took triplicates out every year and analyzed the phylogenetic diversity to try and understand, along with a lot of visual observations, to try and understand how quickly the communities develop under these translucent rocks, and how do they develop? What's the pathway of development? And I'll rush through this quickly because this is a very complex phylogenetic slide, but basically within 2 years, if you look where the big orange arrow is, within 2 years, under marble and the one beside it at the bottom there, the quartz, those are the 2 translucent tiles, you get a surge of cyanobacterial growth within 2 years from the point of initiation. In fact, it's essentially the 3rd year. So even though at 2 years there was very little visual evidence of a hyperlithic community and nothing adhering to the underside of the rock, you're starting to get changes in community structure. And cyanobacteria are the marker organisms because, of course, they're driven by the presence of light and they support the rest of the community. And it's not just one cyanobacterial taxon, there's a bunch of them. I just list those. Those are quantitative figures, some of them with rather large error bars, but nevertheless, the red and the orange lines, those are from the two translucent rocks. The purple and blue are from the controls. The control is samples taken from outside the array, just open soil. And travertine is our black rock control, no light transmission. And you can see there's a surge by 2018, typically in most of them, but not all, of a number of different taxa. Nostoc, Lingvia, well-known. Spirulina is an interesting. Pluricapsa is a common one. So there's a composite of different taxa of cyanobacteria with some different physiologies, we assume, who are contributing to the development of that green cyanobacterial map that you saw. So we developed a sort of a concept of how this operates based both on the visual data that we had and on the phylogenetic data. And typically what happens first, within 24 months or thereabouts, you start to see, you remove the tile, you start to see a green film. You can see it in the so-called soil biofilm phase on the soil under the quartz rock or under the tile, under the translucent rock, but there's nothing adhering to the rock or the tile. Subsequently, it goes through some sort of adherent phase where when you uplift the tile or the rock, there is stuff sticking to it, as it were. And so this has gone from a biofilm on the soil to a biofilm on the lithic substrate. There's quite strong evidence that the development of the biofilm, firstly, is triggered by water addition. That in one of the arrays, we found almost nothing happening over quite long periods. So the one that showed the most significant development was, in fact, the inland array, which gets more frequent water. So the development of the cyanobacterial layers is water-driven. We suggest that the adherent phase is actually driven by dry phases, because one of the responses of cells in a biofilm when they desiccate is to switch on extracellular polysaccharide biosynthesis, and that forms a matrix, expands the biofilm, and that would be the glue that sticks the cells onto the cell. And then after some further period of time, about which we know nothing, you develop into a mature hyperlithon. And one of the key factors was that observationally and phylogenetically, even after 7 years of development, the communities that we could see under these rocks were a very, very long way from a mature hyperlithon that's been just taken out of the soil. So growth, maybe decades. There is a little bit of data from Antarctica that suggests that carbon on the underside of these rocks and growth rates in Antarctica would be much slower, is 300 to 400 years old. So that might be the timescale of a mature hyperlithon. And that, I think I've used up my hour. This has been a wonderfully exciting journey in the Namib Desert with a huge amount of fantastic work, working with a lot of really, really interesting, clever people across a wide range of disciplines. And so with that, I thank all of those who've been part of this program with me over the last decade and a half nearly, and I thank you all for your attention.
Right. Thank you very much, Don, for a very exciting talk and for showing us there's so much life in the desert, even under rocks. So the floor is open for questions. You can show by raising your hand, and then as soon as we acknowledge you, then you can unmute. And let me just also see in the chat if there are some questions. Yeah, so let me just get it started and ask one question. So when you showed that there's some water deep water underground, which can actually evaporate and contribute. How deep is this?
It's we know from the geologists that groundwater depths vary enormously, and in some places they may be a few tens of meters, and some places they may be 100 meters deep. And it's complex because there are different geologies, which may in some places prevent water vapor transferring to the surface. Nevertheless, time is not an important issue here. It doesn't actually matter if it takes a decade or 5 decades for water vapor, because there's a constant supply of water from the subsurface in the groundwater. It doesn't matter if it's 5 decades for it to slowly transition to the surface. So we know nothing about that process and how quickly it happens, but we are starting a series of experiments that might, in fact, give us some clue to that. And this season, last and the next season, we have taken and we will take more samples of water from different places, including groundwater. And we're going to look at hydrogen stable isotope ratios, because with luck, we may be able to track the source of water by comparing it to oxygen in plant tissues, on the grounds that growing plants, the oxygen isotope fractionation data in growing plants will reflect the latest water source they've had. This groundwater, by the way, does come to the surface. There are saline springs, presumably where the groundwater hits a sill of some sort and is forced to the surface. And in fact, there are a lot of springs across the desert. Some of them, most of them coastal, but some of them quite deep into the desert. And we've sampled those as well. And maybe they will reflect the deep groundwater, which otherwise we can't easily get access to.
Yeah, thank you. I see there is a hand up. Robert, you can unmute yourself.
Thanks, Ruben, and thanks, Don, for the presentation. That was fantastic. I'm at Wits University. I study plant physiology, and I'm interested, kind of a general question about the history of the desert. Can you give us some sense of when the desert emerged in time? When did it become the Namib Desert? Thanks.
Yeah, thanks, Robert. It's all I can tell you is what I've read. Because I'm no geomorphologist or geologist myself, although I do know a couple. We see in the literature from authoritative sources that the Namib is quoted as being, well, quote, one of the oldest deserts in the world. The implication being that it has been a desert for many millions of years. I forget the figure exactly. The expert on this would in fact be Frank Eckhart from UCT, and he's published on the fact. But this is an old desert. It's one of the older deserts, and I guess that probably fits with some of the evolutionary processes that we see the results of. For example, things like the Welwitschia mirabilis, an amazing plant. Some of the other species which are desert adapted. Now they occur in all deserts, but nevertheless, there are a raft of very highly adapted species of all sorts, generally macroscopic, not microscopic. So yeah, unfortunately, apart from old, I know relatively little about its geomorphological history.
Great, thanks so much. That was a fantastic presentation.
There's a question in the chat. I'll just read it off. I can't see whose name it is, just a number identifying them. What influence might fossorial animals, including termites, have on below groundwater dynamics?
That's a very nice question, because I've not thought about it.
Sorry, I didn't know that I wasn't identified. Don, it's Tim Hoffman here, and you're probably not surprised that I'd ask about termites. But thank you for a lovely presentation. Yeah, my question is in the room, in the chat. Thanks, Don.
Thanks, Tim. Delighted to meet you even virtually again. It's a really good question. The termites in particular are not a particularly common species in the gravel deserts. They're much commoner in the sand sea. There's no visual evidence of termite activity anywhere in the gravel desert. But then, to be fair, the termites, the sand termites that occur in the sand sea, they don't produce visual surface evidence either. I suspect that the impact on groundwater dynamics is quite low, except for near surface. There's a lot of activity, even in the hyper-arid zone, in the top meter, I guess. Small animals burrowing, and that's going to influence shallow subsurface water transport, but nothing deeper than that. And if the groundwater is 10 meters down, then I think we have to rely on natural filtration processes or evapotranspirator processes to move the water up to a zone where it might get a freer move because of some mobilization of the shallow subsurface soils. Not very satisfying, I suspect, but best I can do.
No, thanks, Don. Difficult to study, for sure.
I think we can take another question if just show by raising a hand and unmute. Okay, if there's none, then I think let's all thank Don for a very nice and exciting talk. It was quite engaging, and let's thank everybody for the very nice attendance and for the very engaging discussion that we've had. As a reminder, before we close off, the next seminar is on the 17th of July. So you will receive notices in due course via email. And also this presentation is going to be available online on the Royal Society's website. Once again, thank you very much, Don.
My pleasure, and thank you all very much for being with me.
Thank you.
Bye for now.